Hello students, my today's topic is origin of life 2, that is prokaryotic and eukaryotic cells. So in case of prokaryotic cells, they are simple unicellular organisms having other features, that is nucleus absent, and whereas other features which are found in more complex cells of all the organisms are called eukaryotes. In 1938, the American biologist Herbert Copeland proposed that unicellular organisms lacking nuclei will be classified into their own kingdom, that is Monera, also called kingdom prokaryotes. All bacteria are categorized in this newly established kingdom. This scheme was first to establish separate kingdom for prokaryotes, that is the organisms without nuclei. Whereas in case of eukaryotes, organism with nuclei. In 1990, American microbiologist Carl Boyce proposed that bacteria will be divided into two groups, archaea and bacteria, based on their structural and physiological differences. In some classification systems, the archaea are considered prokaryotes, in others, they are classified into their own domain. The archaea consists of a small group of primitive anaerobic organisms that do not require oxygen. They are found in a narrow range of habitat, often in extreme environmental conditions with high temperature, high salt, high acidity. In contrast, bacteria live in a wide range of environment with or without oxygen at various temperature and at various level of acidity. This is the structure of the prokaryotic cell. Here nucleus is absent, so we also say here the true nucleus is absent, but it consists of nucleolus and nuclear membrane. Here the genetic material consisted of one or more haploid circular with no free end double-stranded DNA molecule with no associated histone, all the basic proteins. Such primitive kind of nucleus was called nucleoid genopore or prochromosome. Number of genes were found to be less, that is 700 to 6000. Here cell wall is also present, that is made up of peptidoglycan or murine when present, but it is absent in mycoplasma. Peptidoglycan is a polymer of recurring unit of muropeptide. Now what is muropeptide? It is a disaccharide of N-acetyl D-glucosamine and N-acetyl muramic acid, which is interlinked by alpha-1,4 glycosidic bond. In this diagram you also see the cytoplasm, which is very dense, showed no cyclosis. Cell organelles are also present here, that is mitochondria, endoplasmic reticulum, chloroplast, Golgi complex, microtubules. They were absent. Only cell organelles present were ribosomes, which were found to be formed of rRNA 60 to 65% and proteins 35 to 40%, and were of 70S type. So two subunits of 70S ribosomes are larger 50S and smaller 30S. Respiratory enzymes, they were also associated with plasma membrane. Flagellum for locomotion, that consists of single flagellum or fibril with no differentiation into the exoneme and protoplasmic sheath. Here endocytosis and exocytosis were not possible. Cell division occurred only by amitosis, there being no mitosis or meiosis. So, prokaryotic organisms were earlier anaerobe as no oxygen was available in the environment, and probably delivered their energy by the process of glycolysis, which is also found in all present day living organisms. So, this suggests that first prokaryotic cells were probably the ancestor of all the existing organisms. So, this is the characteristic of the prokaryotic cells. That is also the characteristic of prokaryotic cells. Some species of the prokaryotes have a form endospores, thick-walled, dehydrated structure that can resist extreme dryness and very high temperature for long period of time. Anthrax, tetanus, botulism, these are the disease caused by endospore forming bacteria. Certain prokaryotes move independently by using flagella. Here in the diagram I already discussed the flagella. Now the next is the origin of prokaryotes. So in 1862, Pasteur disproved the spontaneous generation theory but left open a question: how did life begin? Miller's synthesis is possible answer, or it may be the seeding of organic molecules by membranes, meteorites from outer space, or a god event that started life. It is generally held that first organisms were formed around 4 billion years ago, with the earliest form being simpler molecular grouping that somehow gained the ability of metabolize or reproduce. It is also held that these simple molecular arrangements formed from existing inorganic substances life from non-life. Now the next is the evolution of prokaryotes. Prokaryotes are mostly bacteria, and their advancement lead to more complex living organisms. Advancement in the structure, function, prokaryotes continued to where two separate types are now indefinable, that is bacteria and archaea. So these are some of the examples of prokaryotic cells. As we say the evolution of the prokaryotic cell, there are various theories. One is called the origin of chemosynthesis. One theory said about the origin of anoxygenic photoautotrophy and origin of aerobic photoautotrophy. Thank you. Please like and subscribe the channel.